This we know. The rate we now know. And the time we know also because we marked down the time. And we can solve for the time that began. And here's what our equation would be. Time that began, here we have it. Subtract the rate times the time from this. K plus the first analysis minus the rate times the time. That's for subtracting this. And that'll be equal to the time that began. So we know the time we can figure out from this K plus ion concentration. After taking it twice, we can find when the body started to die and produce the potassium ions in the eye fluid with this equation. So this is a very good way of finding when the person died. This does depend on the temperature, so it's not as so. But usually, both of these temperatures, both of these samples are taken at the same temperature, and which is pretty much the same temperature of the room that it was found in. So it's really not. So that's really not much of a problem. So this is a very good way of finding when the time the person died. We can also get it from the stomach contents. Depending on the kind of food, it takes a certain amount of time to digest certain amounts of food. So we take a look at the food in the stomach. We try to find out when he ate his last meal, and we say, okay, this is, let's say, it takes 3 hours to digest this. It's 1/3 digested. It must have been it was an hour after he ate. That's when he stopped digesting it. That's when he died. Okay, fine. We analyze stomach contents, but we always do it not just for the food and for the time it took to digest the food, but for the stomach contents because there may have been drugs. We can find undigested pills in there, or maybe some chemicals that imply there were drugs there. And this depends usually not much on temperature, because when a person's digesting, he's pretty much at the same temperature. But some different foods get digested differently, so the analyst has to know what the food is and how long it takes to digest. And all kinds of different foods are different. So this is good enough for, let's say, a couple of hours to maybe a day or two. After that, all these other signs, they stop. The temperature, the alga mortis stops when it reaches room temperature. Liver mortis stops, and reverses itself and by the time it's 36 hours, forget it. You're not getting any information from there. Similarly, rigor mortis doesn't last that long. Doesn't last for more than a day or so. And even the potassium ion concentrate does not lag. Again, you can't figure it out in more than a couple of days. So after that, you have to find out how the body decomposed. So then you have to use the rate of decomposition. This is a little bit less clear how fast it goes and how to trace it, but we can get a basic idea. The body decomposes with what they call autolysis, putrefaction, and we can find adipocere buildup in the fatty tissues. So autolysis, that's when the cell's natural enzymes react. Now, cells have enzymes, the body has enzymes, and they react and they work on what they're supposed to work on because they're controlled. Once at death, the enzymes are still there and they're not controlled, so they react with the cell matter, not just what they are given when the body's alive and the cell is alive. So the body sort of decomposes that way when the cells kind of like digest themselves. And they do it at a certain rate. This depends on the organ, the mechanism of death, or what was exposed, what was not exposed, the position of the body, and of course the temperature. So it's not as easy to find, but there you can get an idea of how much decomposed based on the autolysis of the various organs. That's what the forensic analyst will have to find out, and comes from the autopsy. The other one, the putrefaction, we're more familiar with as rotting. That's when bacteria attack the body. And you can see it, bloating, you see discoloration of the body like purplish or whatever dead bodies, decomposing bodies look like. And it produces a stench. That's the smell of dead bodies. It's been said that the smell of a dead human body is extremely offensive to people more than any other creature. And there was an old question they asked, I don't know how long ago, but it was at least 1,500 years ago. And the question was, called philosophical, why does the human body decompose and leave such a terrible stench? More offensive than any other animal is the decomposing of any other animal is the decomposing of a human being. Why is a person so offensive when they die? And the answer given was, again, a philosophical one. If the body would not be so offensive, people would not bury it. They'd keep it around. And this is not good for the psyche, because if people see the dead bodies of their loved ones constantly, because they kept it around, they're going to get depressed. Better to bury the dead body. But if the person doesn't give off a very offensive stench, people are going to try to leave it around, try to keep their loved one with them, and that's not good. That's why in nature the human body has a terribly offensive odor when decomposing, so that it will be buried quickly and people will not be depressed and they'll be more optimistic about their lives. Well, interesting philosophy here. Let's get back to the topic. Bacteria decompose the bodies. Now, they operate in more fluid areas because bacteria need wetness, they need water. So they get to the eyes and the tongue to swell up, and they eat the tongue and the eyes, and it swells up, so they bulge out often. Again, the skin is kind of dry on the outside. Under the skin, however, it's like, it might be blood, it might be wet. It gets sealed off from evaporating, so it's wetter there. So the bacteria are going to eat up and get all over under the skin. So if you try to pull the skin of, let's say, the hand, you might slip the whole skin off. So it can happen. So again, this is one of the signs of putrefaction and how long it took. It depends on the temperature, availability of bacteria, and maybe the person was already infected with something. And that bacteria just, after the person died and couldn't protect itself against the bacteria, it immediately swarmed all over that area. So it depends. This putrefaction, again, people can tell how long it takes, but it depends on a lot of things, so it's not that easy. It depends on the bacteria in the area. Sometimes there's almost none. For example, they have recovered recently. I don't know how recently, past decade or so, I don't know, but dead bodies in Scottish bogs. These Scottish bogs, had a very basic, whatever it's called, base. They had a very basic concentration. Bacteria couldn't live there. These people, they were killed, and by being thrown into the bogs with their hands tied, and they did not decompose, even though it's been like 1,000 years or so. They just didn't decompose because the bacteria couldn't live there. The water was so basic. And we've recovered these bodies. And again, even the ropes didn't necessarily decompose due to bacteria because there was no bacteria there. So, and we see what these bodies look like. They're, they're non-decomposed. And let me tell you, the look on these bodies is ghastly. They're horrified. These people didn't just fall into the bogs. They were thrown in there either as human sacrifices or for some kind of punishment. Who knows? Whatever it is, the point is that you have to know about the bacteria in the area where the person was killed. Sometimes in really, really dry desert areas, not much bacteria there. They get killed by the sunlight and by the dryness, so the body might not have much putrefaction or decomposition. And fatty parts of the body